Every living being imaginable ages, because the cells that constitute life age. So how can evolution, which enables life to adapt to nature, not overcome this affliction called aging? In this video, we will search for the scientific answer to this question. The place we need to start is understanding what aging is. First, we will interpret this through aging at the cellular level, and then we will address it for more complex life forms. Cells produce energy through respiration, produce energy from nutrients, use this energy to expand their internal chemical pool, and then replicate their DNA and divide when the necessary conditions are met. Fulfilling these conditions constitutes their life. However, cells also age for various reasons during their lifespan before division. Harmful waste molecules produced during energy production, the gradual deterioration and inefficiency of organelles, activation of different gene regions over time in DNA resulting in different proteins, and the accumulation of mutations in DNA are among the reasons for aging before cell division. Also, mitochondria play an important role in this process, because the most harmful molecules are released during energy production here, and mitochondrial DNA is vulnerable to this damage. Moreover, in aging cells, the autophagy and proteasome systems deteriorate over time and become insufficient in eliminating misfolded proteins. These accumulations disrupt intracellular balance and increase toxic effects. Finally, even though the DNA sequence does not change, epigenetic mechanisms that determine when and how much genes will be active also deteriorate with age. This is an important factor causing cells in different tissues to age at different rates. So, does a cell's life end when it divides? Although this was thought for a long time, experiments have shown that aging can also be partially transmitted through division. However, an evolutionarily developed protective mechanism comes into play here. While the cell divides, it copies its DNA, organelles, and internal content to form a new cell. In this process, some signs of aging, such as mutation accumulation or changes in gene expression, may pass to the new cell. However, observations show that oxidative reactants and harmful or misfolded proteins mostly concentrate in one cell, usually the mother cell. Thus, aging continues in only one cell, while the other remains healthier and younger. This provides an advantage in terms of natural selection. On the other hand, division itself carries aging limits. Each time a cell divides, telomeres located at the ends of DNA shorten slightly. Telomeres are structures that protect important parts of DNA, and when they fall below a certain length, the cell can no longer divide. This limited replicability of telomeres determines the total number of divisions a cell can undergo and is known as the Hayflick limit. When telomeres become too short, the cell enters a state called senescence, where it can no longer divide but remains alive. These cells are not passive. They release harmful signals into their environment, accelerating inflammation and the aging process in surrounding tissues. In multicellular complex life forms, aging also continues at the cellular level. However, an advantage is that the energy production requirement can be shared among cells, thus reducing oxidation accumulation. If the organism can eliminate the oxidation accumulated in cells, aging slows down. However, these mechanisms also lose function over time, and all cells in the organism age. For example, in humans, during aging, cellular aging in the immune system increases the frequency with which undetected external and internal threats damage other body cells. In muscle cells, the high energy expenditure accelerates oxidation. And since these cells eventually die, muscle shrinkage begins. Neurons in the brain are the most striking example of cellular aging because neurons cannot divide, so aging effects appear directly. Diseases like Alzheimer's and Parkinson's seen in old age arise from aging neurons due to accumulated oxidative reactants, misfolded proteins, and malfunctioning organelles. As a general rule for all body cells, DNA errors occurring during each cellular division and telomere shortening cause cells to age. Different organisms in nature have different lifespans. Aside from environmental deaths, these lifespans depend on biological aging. We have learned what cellular aging is. If an organism finds a way to slow down the causes of aging, its lifespan can reach hundreds of years. If it finds a way to eliminate these causes, biologically immortal organisms may emerge. Experiments have shown that in the unicellular organism Hydra, telomeres do not shorten with each division, and that it can cleanse harmful substances in the cell and renew itself, meaning it does not age. Some tree species live for centuries. This slow aging results from their highly protected cells, clean energy production, and low energy consumption due to their slow metabolism. Now let's come to this question. Despite the existence of long-lived and even biologically immortal organisms, why hasn't the evolutionary process enabled biological immortality for all species? What we call evolution is the process by which organisms adapt to their environment, eliminate their competitors through natural selection, and ensure the continuation of the species by reproducing and spreading their DNA. If an organism can adapt to its environment, gain an advantage, and reproduce to pass on its DNA, it ensures the continuation of its lineage. After its DNA is passed on, the aging of the organism is no longer of concern to nature because the mission is complete. The DNA has been passed on, and it can now die. In fact, if an organism lives long and retains its reproductive ability, its chance of passing on DNA increases, since organisms carrying its DNA will also reproduce. However, this would create competition for resources, which is a disadvantage in terms of natural selection. Because of this disadvantage, evolution tends to favor death after reproduction in most animals. There are several interesting theories by scientists on this subject. The first is Peter Medawar's mutation accumulation theory, which suggests that aging is primarily due to the accumulation of harmful mutations over time. Natural selection eliminates harmful mutations during youth, but mutations that only manifest in old age go unnoticed and are unknowingly passed on to the next generation, since individuals have already reproduced before these mutations become detrimental. Another theory is the Antagonistic Pleiotropy hypothesis, which suggests that certain genes provide advantages during youth and reproduction, but have harmful effects later in life, after reproduction, thereby preserving the species' advantage. Lastly, there is Thomas Kirkwood's Disposable Soma theory of aging, which argues that in order to be evolutionarily advantageous and reproduce, cells allocate most of their energy and resources to reproduction and survival until reproduction, while dedicating fewer resources to repairing cellular damage that causes aging. This behavior provides an advantage in natural selection but makes aging inevitable. As can be seen from these theories and the nature of evolution, nature only supports living long enough to reproduce and pass on genes. It does not take any steps regarding life afterward. In other words, natural selection chooses to live long enough to reproduce, and preventing aging has not even made it to the shortlist. That's it. Although evolution may not see aging as a disadvantage, we humans know how undesirable it is. Who knows? Maybe transhumanists will develop enough technology to eliminate all flaws of the human body, and all humans will live forever, turning the world into a hellscape. Until the next video, see you. And remember, everything comes from science.